Fluorescence imaging is a powerful tool in understanding cellular function and makes use of dyes to detect specific organelles and structures which play a role in cellular function. Cellular stains act as counterstains to identify the location of organelles and as reporters for changes happening in the cell. Cellular stains from Biotium can be used to probe the location of specific proteins and organelles. We carry a full range of cellular stains that include membrane stains, nuclear stains, and stains for a wide range of other organelles. These stains are useful for live cell or fixed cell imaging, as well as for flow cytometry. So how can you find the best stain for your application? Let's consider how the workflow will affect the choice of stains and how to navigate the resources we have available to make this selection. Different dye characteristics are important when staining live cells versus fixed cells. For all workflows, the dye must reach the organelle and be specific to that organelle. When staining live cells, however, the dye must also be non-toxic. If the stained cells are to be fixed, the dye must then withstand fixation, and if the cells are to be permeabilized, the dye has to tolerate detergent. In fixed cells, the dye might reach organelles more easily, but will need to have specificity for that organelle. There are a lot of resources at the Biotium website to help with dye selection. Let's see an example of how the workflow impacts the selection of a dye to stain the cell membrane. Biotium carries a number of dyes to stain the cytoplasmic membrane. Two of the lipophilic membrane and cell surface stains are Cell-Brite cytoplasmic membrane dyes and Cell-Brite Steady. Cell-Brite cytoplasmic membrane dyes are dye delivery solutions that can be added directly to normal culture media to uniformly label suspended or adherent cells in culture. Cell-Brite dyes primarily localize to the cell surface membrane shortly after staining. However, in live cells, the stained membranes become internalized by endocytosis, so staining becomes primarily intracellular over the course of a few hours in culture. Cell-Brite Steady was developed for long-term staining of live cells and allows fluorescence imaging of the cell surface for up to several days in culture. The Cell-Brite Steady dyes are unique fluorescent membrane probes that distribute between cell surface and intracellular compartments, so cells retain cell surface staining over time. With the use of Cell-Brite Steady Enhancer, intracellular staining can be reduced or eliminated for imaging of cell outlines or boundaries. Cell-Brite Steady has also been validated for exosome labeling and comes with storm-compatible dye options. The lipophilic membrane stains have poor tolerance for permeabilization or methanol fixation, so Cell-Brite staining is not easily combined with intracellular immunofluorescence staining. Membrite Fix and Cell-Brite Fix are covalent stains that can be fixed and permeabilized for intracellular immunofluorescence staining. Cell-Brite Fix membrane stains are fluorogenic reactive membrane dyes that rapidly accumulate at the plasma membrane. When they incorporate into lipids, they become fluorescent, and at the same time react covalently with membrane proteins for stable labeling. Membrite Fix requires a two-step staining protocol with washing, but offers a more extensive choice of dye colors than Cell-Brite Fix. Membrite Fix can also be used to stain yeast, but unlike original Cell-Brite dyes and lectins, Cell-Brite Fix and Membrite Fix staining must be performed in live cells and fixed shortly after. We also carry CF dye lectins with a wide choice of CF dye colors. Lectins work with all workflows. However, staining varies with cell type. In addition to workflow, there are other things that must be considered when selecting the best stains for your experiment. Microscopy and flow cytometry have different requirements for a dye to be optimal for its use. Microscopy benefits from photostable dyes, while flow cytometry data is improved by using brighter dyes. Biotium carries a variety of dyes and kits optimized for these applications. A useful resource when selecting cellular stains is the Spectre Viewer, which can be found at our website. The Spectre Viewer allows you to select excitation sources or filters, emission filters, and dyes. This allows you to see if the dyes selected will work together and will work with your instrumentation. Biotium carries dyes in a wide range of colors, making multiplex staining easy. When designing experiments, our technology pages provide an overview of the technology and compare different products we offer with links to the product pages to help make your selections. These can be found under the Technologies menu. For instance, when selecting cytosolic membrane stains, there are a few pages at the website that are useful. The Cellular Stains Comparison Guide and the Cell Surface Stains Technology page both have a lot of information helping you determine which stains are best for your application. We also have brochures in the brochure library. These technology pages have information on which stain is recommended for different workflows, comparison tables with dye characteristics, the pros and cons of each dye, and the staining characteristics in different cell types such as bacteria, yeast, and mammalian cells. These are additional links and resources that can guide your experimental design. We have technology pages with an overview of cellular stains that has an intuitive interface to find the options for each organelle. There's also a technology page with stains that have been verified for exosome and EV labeling. Also note that the table which will compare which organisms are labeled with different stains is in the microbiology section of the website. We have a lot of additional resources with product information on our website. There's a list of frequently asked questions about our products that is updated regularly. There are additional tech tips and protocols, our brochure library, publications, and highlighted citation. These can all be found under the Support and Resources section of the website. Protocols for kits and dyes developed by Biotium can also be downloaded from the product page. If you still have questions or need additional guidance, we encourage you to reach out to our tech support team. This is an overview of some of the stains and dyes we carry for visualizing cellular structures, in addition to the membrane and cell surface stains. We also carry a full array of antibodies labeled with our bright and photostable CF dyes, fluorescent proteins, or haptens, and a variety of kits to measure cellular processes such as apoptosis, cell viability, vesicle trafficking, and so on. I've shared some of the resources that can be used to make a selection. Biotium was founded in 2001 and is a leading life science reagent manufacturer and supplier. We are devoted to providing high-quality and innovative fluorescent tools that fuel scientific discovery. Our collaborative team of experienced chemists and biologists who are at the forefront of fluorescent dye design apply chemistry-based principles toward producing solutions for unmet challenges in life science and medical research. We pride ourselves in providing extensive information on our dyes and their use, and expert solutions and answers to your questions. At the end of each tech support line, there is a PhD scientist ready to solve tricky problems and respond to customer inquiries. If you are ready to turn your hypothesis into discovery with Biotium's novel cell stains, head on over to the website to download our Cellular Stains brochure and the Cell Stain Selection Guide. All of the links shared in the video today are in the description. Thank you for listening.